speaker is Aviv Avraham Mezer, a new approach for reproducible water fraction and T1 mapping across different qMRI protocols. Aviv, please.
Thank you. So I'm going to present here on behalf of Eden, a student in the lab that couldn't arrive, and this work was done in a collaboration with José March from the Netherlands. And I'm going to talk about T1 mapping and particularly on water fraction mapping. And this is my, how do I move the slides? This one? Okay. Okay, and this is my disclosure. Okay, so T1 is a relatively used quantitative measurement, observed T1, that used to look on a microstructure in the brain, and particularly in myelin. And one of the main methods to do that is variable flip angle, in which we use at least 2 flip angles, and then we fit the single equation, and this allows us to estimate T1 and another image called M0. And another common method is MP2 range, in which we typically use at least 2 inversions, and we can fit the single equation again, estimate T1, and again another image, which is the M0. One advantage of variable flip angle is that we can use this M0 to estimate water fraction. In this approach, we first fit T1 and M0, as I said before, and then we adding a post-processing analysis in which we regularize the water fraction estimation and remove the bias inhomogeneity and estimate in the end a PD map. And then the last bit is to calibrate essentially the PD map to be water fraction, and this is done using calibration with the CSF, which gives us another quantitative map. So now we have 2 maps, T1 and water fraction, which are good to have together, and there's more modeling that can be done when we have 2 maps. So today I would want to briefly explore the differences or the agreement between the 2 T1 methods approach to estimate T1, and then also show how we can estimate water fraction with using both methods and show their agreement. So let's start with the T1 mapping and agreement. So what we see here is a 2D histogram, and the dark color here represents a number of voxels, and we see here scan and rescan. So with invariable flip angle, we have a very good agreement. So it's actually quantitative. And this is also true for MP2 range. You can see in both methods, we have a very good agreement when we scan the same subject twice. So we have a good quantitative maps. And the question if it's true also between methods, between protocols. And so we test that, and it's actually pretty good. Not exactly as it was before for each method, but it's highly reliable. That's what we see here. And we can also look at that in the level of the single subject. So here is the map of each, of some subject of this 14 subject, and you can see it agrees in any subject. Okay, so T1 seemed to be reliable and useful, and both methods should feel good about the fact that our quantitative method is actually quantitative. Next, I will say something about water fraction. So I mentioned before the pipeline in which first T1 and M0 is estimated, and then there's a post-processing to estimated water fraction, and that was for variable flip angle. And the question is, what about MP2 range? So we actually applied the same approach, and here you can see that we can estimate proton density, and now the last part is estimating the water fraction, and it's slightly harder because the CSF estimation in MP2 range is not as good. So we had to adapt the method, and I will say a few words about that now. So that's some innovation here. So the approach is as follows. We first identify the ventricles, and we identify CSF voxels by the T1 values, and also omitting values which are just outliers. We then fit a linear regression essentially between 1 over T1 and 1 over PD estimating two parameters, two coefficients here, B1 and B0. And having that, we can add a known value of T1 in CSF, in pure CSF. It's supposed to be, the T1 is supposed to be 4.3 seconds, so we can actually plug it into the equation and see what PD we have. So we have all the parameters here. And so we have some value of PD, and we can now find this single parameter, this single coefficient that will allow us to make PD of the pure CSF as 1, and then we have a true water fraction map. So that's what we do. We just then use this calibration parameter to create a water fraction map. Okay, so this is the reliability of this approach to estimate a water fraction with variable flip angle. As you can see, it's pretty high. It's not as good as T1, but it's relatively high. This is scan-rescan of 3 subjects. And this is for MP2 range. Also highly reliable, and this is important because now we can estimate water fraction also with MP2 range, and this is new. And if we compare between the methods, you can see that we also have a relatively good agreement that the values are on the identity line, although they are not exactly 1-to-1. And this is in the single subject level here. And as I said before, it's reliable. We have some cases in which we have some offset, like in here, which we need further research, or just we have to live with that they are not exactly perfect. So to summarize, we saw that T1 mapping is actually highly reliable using these 2 different quantitative methods and can be comparable. We then look on water fraction mapping, and we saw that we can actually estimate in both with variable flipping and with MP2 range. And we saw they agree very well as well. And with that, if you want to hear more, you're welcome to come to the poster. I'm actually going to alternate between these 3 posters. The other 2, one is about semi-quantitative methods above and beyond T1 divided by T2. And the third one is about new biophysical model of myelin packing. And with that, I will end. I will say thank you to Eden and to Jose, and thank you.